drowning, and through a succession of peristaltic waves propel the contents into the stomach. There isn't much secretory activity going on in the esophagus. However, the esophagus does have mucus glands, and there is a degree of mucus secretion from the submucosal. We also mentioned that the esophagus is characterized by the presence of skeletal muscle toward the UES, the upper esophageal sphincter, and smooth muscle toward the LES, the lower esophageal sphincter. We pointed out that there is an abrupt transition in the epithelium between the esophagus and the stomach, characterized by the Z-line that generally occurs at about the cardiac or cardial stomach. That transition is from predominantly squamous epithelium to predominantly columnar epithelium. And we'll talk more about that columnar epithelium and its organization in the stomach as we continue. So we talked a little bit about the remarkable ability of the stomach to distend without dramatically increasing intragastric pressure. And that in part is made possible by the process of receptive relaxation. Remember, the stomach wall has 3 layers of muscle. The muscle is fairly tenuous. It's fairly difficult to stretch that muscle. However, this particular muscle has more or less an atonic, relaxed state. And as a consequence, the stomach can expand, can distend considerably without increasing intragastric pressure, as might be expected, for example, if the stomach were more like a balloon. Now, we've already defined the major types of motility. We've talked about peristalsis, mixing, and so on. Certainly in the stomach, we indicated that the superior or upper stomach, the body of the stomach, is primarily important for acting as a reserve, acting as a storage chamber for ingesta. And that the distal stomach, this antral or pyloric part, is where most of the churning or mixing occurs. The rate of peristalsis is on the order of maybe 3 or 4 per minute, typically, so very, very slow. And as in the intestine, the smooth muscle, as you remember, is myogenic, but in the GI tract, especially in the intestine, but to a lesser extent in the stomach, the smooth muscle contractions seem to be regulated or timed by these cells of Cajal, interstitial cells of Cajal. These are arguably analogous to the sinoatrial node of the heart. And these present a bit of a problem physiologically because the smooth muscle is myogenic, yet the frequency of contraction seems to be organized around the pulses generated by these cells of Cajal. We pointed out the antral stomach generally consists or utilizes a motility wherein in the antral region the chyme is cycled back and forth, ultimately for the purpose of allowing small amounts of chyme to pass into the duodenum via the gastroduodenal junction. And we need to talk about secretion. We'll do that in a moment. The stomach mucosa is lined with gastric glands, and those gastric glands perform a variety of functions, but there are a number of specialized cells we'll look at in just a moment and talk about the secretions, some of which you see are listed here. The stomach initiates, or arguably initiates, the primary process of digestion. There is some digestion going on in the mouth, salivary amylase, salivary lipase, and so on, but those are fairly weak enzymes. Digestion in the stomach is achieved by a variety of chiefly proteases, and these proteases go to work on at least a portion of the protein. Normally we say that about 20% of dietary protein is digested in the stomach, the remainder in the proximal intestine. Yeah.
So if someone has gastroparesis, would you say that affects mostly the lower 1/3 of the stomach?
Well, I think in that case it could occur anywhere, but I think the physiological reduction in function would be most problematic initially. Because again, this region is not moving much anyway. So it's going to be this region that does does most of, or is responsible for most of the motility, and we said that most of that is focused upon regulating the amount of chyme that moves to the intestine. And that's why when we talk about the gastroduodenal junction, you should think about regulation, allowing the chyme to pass through that canal, through that pyloric sphincter, at a frequency and at a rate that is appropriate for digestion, allowing digestion to occur. Oh well. So that process of passing the chyme from the stomach to the duodenum is called gastric emptying. It occurs at this gastroduodenal junction. Goes by a variety of names. We won't be too concerned about that. As you know, I generally prefer the physiological term, so we just call it gastroduodenal junction. And the chyme is admitted very, very slowly in response to three primary criteria listed here: the composition, the acidity, and the concentration. By concentration, we mean primarily the osmolality of the chyme. So composition, we're talking about the ability of the stomach, and for that matter, the intestine, to preferentially digest some substances more readily than others. For example, fats and proteins are not easily digested, so they take a little longer, they're more refractory. Carbs and aqueous solutions tend to be passed rather readily. Needless to say, the pH, the very harsh acidic pH of the stomach is allowed to pass too rapidly. That chyme is allowed to pass too rapidly into the duodenum. That could cause ulceration in the duodenum. So naturally, the lower the pH of the chyme, the slower the rate of gastric emptying. And then finally, by concentration, what we mean here is that chyme of very high concentration, chyme of very high osmolality, tends to be passed much more slowly than more neutral or even basically chyme of basic acidity. Now we're going to talk about the agents responsible for digesting the chyme. But keep in mind, most of what occurs in the stomach is just storage and initial digestion of the protein. Now this little animation, before we get going, talks about the three phases of gastric secretion. Understand that these three phases also apply to gastric motility as well. So we talk about the three phases. We're going to talk about the cephalic, the abdominal, and intestinal phase. Bear in mind that really the terms apply across the board to all aspects of motility and secretion. Oops, I guess that's why the other one didn't work. Yeah.
Secretion is divided into three phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food sends nervous impulses to the medulla oblongata. These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsin in the stomach. The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach. This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsin secretion in the upper and middle parts of the stomach.
So note that even in the cephalic phase already, you have an autonomic pathway, you have an endocrine pathway. Be sure you understand this gastrin is a hormone released from those gastric pits into the general circulation. And what is its target? The stomach, rather paradoxical.
In the gastric phase of gastric secretion, food has entered and distended the stomach. This distension activates a parasympathetic reflex via the medulla oblongata and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsin. In the intestinal phase of gastric secretion, chyme has entered the duodenum, so gastric secretion is no longer needed. When the chyme contains lipids from the digestion of fats or contains enough hydrochloric acid to bring its pH below 2, gastric secretion is inhibited. The lipids and hydrogen ions inhibit gastric secretion by 3 simultaneous actions. First, they cause impulses to go to the medulla oblongata to decrease parasympathetic stimulation of the gastric glands. Secondly, they set up local reflexes via neurons in the wall of the gut that decrease gastric secretion. Thirdly, they cause the release of 3 local hormones which travel via the circulation to the gastric glands and inhibit their secretion.
But more fundamentally, make sure you understand that this is a signal that originates in the intestine, originates in the duodenum. And unlike the gastric phase, the target is not the same tissue that released the hormone, rather it's the stomach. So we saw in the gastric phase, it's the stomach signaling itself, so to speak. In this case, in the intestinal phase, and really all we mean by that is the chyme is now moved into the intestine. The signal is from the intestine back to the stomach. So keep that in mind, especially as we continue our discussion of these hormones that are secreting GIP and CCK. Now, the mucosa of the stomach is characterized by these invaginations or convolutions or folds that are called gastric pits. So the glands of the stomach mucosa are pits. And you can see these pits here. They're usually tubular, sometimes they're branched. And here you have an idealized, simple little tubular gastric pit gland. And let's talk a little bit about some of the cells that are present. Now remember, the pH is very, very low out here. It's very, very harsh, very, very corrosive in this region. Consequently, it should be no surprise that you have a variety of cells that are specially designed for replenishment. These are cells that basically, as the soldiers die, they replace them. So they're very, very actively mitotic. We're going to talk about a barrier that resides between the mucosa of the stomach and the lumen, called the mucous bicarbonate barrier. But in any case, in order to generate and sustain that sort of viscid mucosal film above the epithelium here, these mucous neck cells are utilized to pump out the mucus at a fairly high pH. All right, as we move down more deeply into the pit here, we encounter a profusion of these cells that go by a variety of names. For the most part, we'll just call them parietal cells. They're also called excentric. They're also called tubulovesicular cells because of how they change conformation when they become activated. But these cells are most important for generating hydrochloric acid. So the most important, well, I got to be careful here, I shouldn't say the most important function, but one of the important functions of the parietal cell that lines the gastric pit is secretion of acid. And we'll talk more about how that occurs in just a moment. The other thing they release, though, is something called intrinsic factor, and we'll return to that. But intrinsic factor is necessary for uptake of B12. In its absence, patients suffer pernicious anemia. You see a number of cells here sort of colored purple. Those are called chief cells. They get their name from the fact that they're the most abundant cell in the gland. And they release zymogens. Zymogens are preliminary enzymes. So these are pre-enzymes. We've already indicated in the stomach most of the enzymatic activity is proteolytic. So these are little secretions that eventually become proteases that begin the process of breaking down dietary protein. And then finally, there are actually other cells. There's some other cells present in the gastric pits, but the only type that we need to be too concerned about are these enterochromatin-like cells. Pterochromaffin. The chromaffin just means that in histological preparations, they're fond of stains. They take up stains fairly readily. They have an affinity for the coloring of the stain. And these are primarily endocrine cells, and we can see some of the hormones that are released over here. We saw in the animation that gastrin is released. Gastrin promotes release of stomach acid. This might surprise you. We've encountered these cells before. Where have we seen these cells before? Yeah, the pancreas, the brain, they're in the liver as well. They're all over the place. Sometimes we know what they do. Sometimes it's not quite clear. They seem to be important in moderating or suppressing G protein activity in some locations, but not always the same way. And then ghrelin is another hormone with which you should be familiar, although we won't talk about it much in here. Ghrelin is an abdominal hormone that plays a very important role in regulating appetite. Not the only one. There are probably some 30 different hormones that, in one degree or another, regulate appetite. Everything from fat cell secretions, leptin, and so on, to a variety of peptide hormones that are released in the brain, especially in the hypothalamus. So be familiar with this range of cells that are present in the gastric pits of the stomach mucosa. And by the way, as you look at the illustration of the mucosa here, notice that all of this is the mucosa. Here is our external muscularis mucosa, and then deep to that would be the submucosa, and there might be some glands there as well. So why is the stomach so acidic? A couple of theories put forth. Anybody have any strong feelings on why the stomach is so acidic? Would you want it otherwise? You can take the Tums and Rolaids and various medications to buffer the pH of the stomach. Sometimes it can be problematic to get heartburn. In fact, the most common symptom that is mistaken for heart attack is heartburn. And you have to be careful in your line of work because there are a lot of people who are diagnosed with heartburn who actually have legitimate cardiac issues. But in any case, what is the function? What is the purpose of the low pH in the secretions of the stomach? I think so, yeah, the opposite activate enzyme. So make sure you understand the function of the low pH in the stomach is not primarily to act as a corrosive agent. Rather, what's going on in the stomach is that this very low pH abdominal juice, this gastric juice, is activating those zymogens, those pepsinogens, into an active enzyme, which is pepsin. So anytime you see OGEN at the end of a word, that means it's in an inactive preliminary state. And when that substance, that pre-peptide is activated, then you generally lose that suffix. So in order for these proteases to reach their maximal functionality, the pH has to drop below 3 or so. And at that point, they can go to work on the dietary protein, and as we've indicated, begin digestion of up to 20% of it. But the gastric juice contains a variety of substances, including, of course, mostly water salts, hydrochloric acid, the pepsins, lots of mucus. We'll come back to the mucus in just a moment. And then this intrinsic factor, which we said is indicated or is necessary for uptake of vitamin B12. So here is a very simplistic representation of these parietal cells. And some of this should look fairly familiar to you. Make sure you understand at the bottom here, this is the blood, this is the basolateral surface here, this is the apical surface up here, and so the top is going to be the lumen of the stomach. And so, what is this? Seems like that's fairly familiar. Where did you encounter this before? Yeah, this is our good old buddy, the carbonic anhydrase reaction. So as long as carbon dioxide and water are present, there's going to be rapid catalysis into eventually bicarbonate and acid. Now, pay attention in the stomach which way those two components go. So for example, the acid that's generated by the carbonic anhydrase in the parietal cell goes where? It goes to the blood or goes to the lumen?
Lumen.
All right, so make sure you note here's the acid here, leaving in exchange, at least partially in exchange, with potassium. And that's an active transporter. In fact, this is one of these rare circumstances in which both are moving against their concentration gradients. Now, the bicarbonate, on the other hand, is leaving for the blood. So the bicarbonate is leaving for the blood, and what's being exchanged with the bicarbonate? We saw this in the lungs as well. Chloride. This is the chloride shift. Bicarbonate for chloride. The chloride concentrations build up in the cell, and consequently, it should be no surprise that chloride can be passed through the apical surface by simple passive diffusion, passive transport, I'm sorry. And so it passes right on through the membrane there. So be sure you pay attention to the mechanism by which hydrochloric acid is secreted into the lumen of the stomach by the parietal cells, primarily by means of our carbonic anhydrase reaction.
Hydrochloric acid is produced in parietal cells through a complex series of reactions. Carbon dioxide diffuses into the parietal cell, and the enzyme carbonic anhydrase catalyzes a reaction between the carbon dioxide and water to form carbonic acid. Carbonic acid dissociates into bicarbonate ion and hydrogen ion, and the bicarbonate ion is transported back into the bloodstream. An ion exchange molecule in the plasma membrane exchanges bicarbonate going out for chloride coming in. The hydrogen ions are actively transported into the duct of the gastric gland, and the negatively charged chloride ions diffuse with the positively charged hydrogen ions. Potassium ions are counter-transported into the parietal cell in exchange for hydrogen ions. The net result is production of hydrochloric acid in the parietal cells and its secretion into the duct of the gastric gland.
As you look at that, you might be contemplating some mechanisms by which this might be pharmacologically arrested. If you have a patient with excess stomach acid or some other malady associated with the release of too much acid into the stomach lumen, how might it be corrected? What could you do? What type of drugs might be useful in addressing that? Now, here we have a very simplistic representation of the stomach, and what we're looking at here, what are called hydrochloric acid secretagogues. A secretagogue is simply a substance that promotes secretion, and so these are substances that promote release of hydrochloric acid from the parietal cells of the stomach. Now, there are three listed here. There are actually many more, but these three are frequently the targets of a variety of drugs, so they're worth perhaps noting in this context. Histamine, acetylcholine, and gastrin. We saw gastrin in the animation previously. Now, these are listed in order of potency. By that, we mean these are listed in the order with which they promote secretion most readily. However, you should know that gastrin is by far the most abundant of the three, and therefore probably has the greatest overall effect, even though it's least potent. So let's look at some of these pathways. I think you're probably familiar with some of these. For example, this cholinergic pathway, right? How do we look at this one here? What's going on there? Does that pathway look at all familiar to anyone? The one that's labeled right here. We have a synapse there going to the parietal cell, to the mucosa. Does this give you any hint? Yeah, parasympathetic rest and digest. So parasympathetic activation, we're just looking at the preganglionic and postganglionic neurons of the parasympathetic pathway. And that, consistent with the job of the parasympathetic nervous system, is going to promote release of acid, promote secretion, promote digestion in the stomach. There's another pathway here that's a little more complicated. The terminology has actually changed recently. But here's the parasympathetic nervous system synapsing with another neuron that actually promotes release of this substance, gastrin-releasing peptide, GRP. And you recall gastrin is a hormone. So gastrin is released from those enterochromatin cells into the general circulation, travels all about the body and binds to, surprisingly, receptors in the mucosa of the stomach. Once again, promoting or enhancing release of acid. So a couple of very different mechanisms by which this can occur. But as I say, they've actually changed the name of this, but I think the principle is about the same. Let's see what this is. All right, I asked you to think about ways that acid production can be inhibited. Let's look at the first here. So here's our tubulovesicular cell. By the way, this parietal cell is also called a tubulovesicular cell because it changes shape and conformation as it becomes activated. Histamine binds an H2 receptor. Here we have gastrin binding a receptor. And what all three of these do, they promote release of hydrochloric acid from the parietal cell. And then you can see the acid being released into the lumen of the stomach. But if you heard, probably second only to Viagra, or at least at one point, was the proton pump inhibitors in the reduction of stomach acid. And so it doesn't matter if you have the other stimuli present, the other agonist present, as long as there's no ability of that parietal cell to release acid, these have no effect. So very effective therapy, but there are some problems with the proton pump inhibitors. They've been shown to have all kinds of potential side effects. And as usual, I broke my presentation again. What happened there? Anybody want Windows 10? Anybody have? Anyway, I will, if any of you Apple enthusiasts want to have a race in terms of booting, how long it takes to boot, I'll happily challenge you there. Just what you get when it boots up may not be what you wanted, is the only problem with this game. But on this thing, it takes about 12 seconds to boot the operating system. So here are parietal cells, or at least this is the mucosa. This would be the outer margin of the gastric gland. And recall out there we have the mucous neck cells, and these are the mucous neck cells arranged more or less peripherally along the gastric mucosa. And they're releasing a couple of very important things that are necessary for all of these glandular cells to survive. They're releasing copious amounts of bicarbonate and mucus, thereby generating this sort of, well, it's called a mucus bicarbonate barrier, but it's a thick viscous film of mucus along the surface of the glands. What's the pH inside these mucous neck cells in the cytoplasm? What would be the pH in there?
Very low.
Any guess? What's the pH out there? What's the pH? It tells you up here. What's the pH out here in the lumen? Yeah, very, very low. It's like a carbonated beverage or something. So what is it here? What? So you're thinking like carbon dioxide generation. If it's 2 there, it must be even lower here. No, it's about pH 7 here. It's about neutral pH. So you see the problem. What if that corrosive acidic gastric juice were allowed to come in contact with these delicate glandular cells, the cells of the gastric pits? It would be, you eventually get some type of potential ulceration and so on. So this barrier, illustrated here, is sort of a yellow yellow film overlying the cells is protective and serves to buffer that transition. Again, it's a pH gradient from approximately 7 to 2. Is this environment compatible with any type of life at all? Can anything live in the stomach?
I'm sure.
What could live? What? That brings up the whole question that you're gonna have in your clinical courses about what fluids in the body are sterile. I think you might be surprised how recent science has changed all that. Yeah. So there are bacteria, at the very least, Helicobacter, which in my lifetime was a bit of a remarkable discovery to the extent that it was found to be the principal cause of stomach ulcers. Great deal of objection, great deal of hostility to that notion. But if it's true, when I asked you a while ago, what is the function of the very low pH in the gastric and the stomach? One of the answers you sometimes read in the textbooks is it's a simple immunological defense, preventing whatever you put in your mouth from infecting the rest of your body. Something to think about, but in any case, there are bacteria that can survive the very harsh environment of the stomach. Now we've talked a little bit about the functions of the stomach, acting primarily as a storage tank, beginning the initial digestion of protein. So, I guess my question for you, a lot of questions here. Do you need your stomach? Can you live without your stomach?
Mm-hmm.
Do people live without their stomach?
Yes.
Is there anything in the stomach that is absolutely indispensable, which if you removed it would dramatically reduce your risk of your likelihood of survival? What is that thing we just talked about? Intrinsic factor. So if individuals, for whatever reason, let's say cancer or perhaps bariatric surgery, lose significant portions of the stomach, they nevertheless have to take not so much intrinsic factor, but probably supplemental B12 to compensate for the inability to take up. Now, one of the more remarkable findings is that individuals who undergo the bariatric surgery for weight reduction purposes, is everybody familiar with stomach stapling? I think we looked at the procedure and so on. There are very different, at least a dozen different varieties of this surgery. There's a brief discussion here, banding, gastrectomy, bypassing. You can actually take the esophagus and tie it to the duodenum there. You can wall off portions of the stomach. There are all sorts of variants of this procedure. But one of the most remarkable findings is that in many cases, at a very high percentage of cases, it corrects the type 2 diabetes that was associated with the condition. In fact, as a PA, you're going to be giving patients advice. What is the optimal therapy for type 2 diabetes now? What is recommended? Diet, lifestyle changes, no longer supported by the evidence. Do you know what the most efficacious therapy is? Superior to lifestyle and dietary changes? Yeah. They've done several studies. So whether that means you're going to ask every type 2 diabetic to undergo bariatric surgery is a different issue. But it does seem to be the most effective.
I thought that when they did that, they switched around the duodenum.
Yeah, you can do it. As you can see, the illustration on the right, there are all number of variations. In the most recent study that I looked at, they divided the patients, the experiment, into 3 groups. 2 different types of banding procedure, which are more like what we're looking at over here on the left. It wasn't even a complete bypass. And I think in one group where there was a minimal intervention, the reduction or the improvement in insulin to glucose ratios was on the order of 20%. In the group that had the more severe operation, it was on the order of 60% improvement. So what that would suggest is just most any intervention seems to be helpful. Of course, the question is why?
So if that's a recommendation for
Well, I'm not saying it's a recommendation, I'm just saying that's what the science
They're
shows.
saying, okay, it's just, I thought the requirement before was you could be 100 pounds of the weight, or it was at one time, at least 100 pounds of the weight.
But see, that's the issue. What you're going to be learning about in your clinical courses are first-line therapies, optimal treatments, and so on. They have various terms to describe that. But if indeed, that's my question for you, is what should govern the therapeutic recommendations that you make to patients? Should it be history? Should it be something written in a book, in a textbook 30 years ago? Or should it be current science? I don't know. I'm always presented with the dilemma of whether I should give you information that you're going to encounter in your licensing exams or information that's actually accurate. And sometimes they're not always consistent.
Licensing.
But I guess what I'm suggesting is it wouldn't surprise me if that recommendation changed. I know, let's say, I guess it was about 12 years ago, when I was on the main campus, there were two pharmacy students, OBs, who underwent this procedure. One did not survive, the other one had suffered some long-term, very ill health effects. In fact, at that point, the survival rate of that procedure was only about 93, 90%. That's dramatically changed now. So I just think it's a complex issue, and it's something you want to think about. That, by the way, that most recent study just came out in July of this year.
I'm just very surprised because my mom actually had the surgery 12 years ago, too, at the same time. And you undergo a crazy, preliminary test that goes to, psych evaluation and make sure you can handle, and, any kind of, I don't know. It seems like it'd be too easy to recommend off the bat.
I know, I agree with you on that point, except again, obviously, because I knew both of those students, worked with them several times a week. But at the same time, I had a niece who went into the hospital. She was heavy too. Done, no big deal. But she had one of the stapling procedures when they walled off about half of the stomach. So the standard seems to be changing on that level too. I don't know. I'll leave that. You folks are I'll let you sort all that out. But I do think it is interesting. Now, again, getting back to basic science, can anybody explain that? What on earth would the walling off the stomach or bypassing the stomach have to do with type 2 diabetes? Anybody have any theories, suppositions?
Sugar won't be digested as much. It doesn't go to the stomach. If you really just get
Well, again, there's really no carbohydrate digested in the stomach anyway. But not the only digestion that occurs is.
Do some of these hormones play a role in insulin resistance?
That's the question, and I think you hit upon it. Yeah, I think that Bob is talking about the hormones. I think the supposition now is that there probably are some substances that we don't thoroughly understand, or substances that we haven't discovered yet that probably contribute to this response. But there are a number of theories out there, but I don't think any of them have been well supported by the current evidence. Nevertheless, the empirical result is quite surprising and impressive. All right, let's make our way into the duodenum. We're looking at the submucosa. These are intestinal villi here, so we're in the intestine now. No longer are we talking about gastric pits, we're talking about intestinal glands, and they have an even more bizarre name. But before we begin talking about the intestinal glands that would be present in the duodenum, and again, you can see little finger-like projections that emanate from the mucosa here. These are the Brunner's glands that are present in the submucosa. They're only present in the duodenum, and their function primarily is to increase the amount of higher pH mucus that is secreted. Why would there be large amounts of mucus at a high pH released in the duodenum? What would be the function? To buffer the gastric juice. And we talked about the gastric emptying and how important that is. All right, here's a sort of idealized transection of the intestine. We can see these little projections here. Those are analogous, I suppose, to the ruga of the stomach. The ruger were the convolutions that allow for receptive relaxation in part. These are called plicae. I think we talked about these in the anatomy. These are the plicae circularis. Go by a variety of names, bowels of conaventes, Kirkring, and so on. And then extending from those are the villi. So those little finger-like projections projecting from the plicae are the villi. And then what you can't see, suffusing the villi, is the brush border or the microvilli. The villi are just barely visible at a native eye resolution. They look like little papillae on the surface of the intestinal lumen. And in the anatomy, we found, for example, that the density of villi tends to decrease as we move from the duodenum to the ileum. Eventually, remember, there are no villi in the large intestine. But conversely, the amount of mucus secreted into the GI tract tends to increase as we move further along. We talked, for example, today in the anatomy about the pectinate line and those anal columns that secrete mucus even in the colon. All right, so the regulation of contraction in the intestine is also orchestrated by those interstitial cells of Cajal that seem to time the rate of peristaltic contractions. Notice that the native rate is much higher than it is in the stomach, on the order of 12 per minute, as opposed to 3 or 4 per minute in the stomach. But that rate tends to decline as we move from the duodenum to the ileum, from about 12 or so to about 7 or 8 per minute. We saw in a previous slide that the autonomic nervous system affects more the amplitude than the frequency of contraction, but in reality, the intensity and the timing of motility in the intestines is affected by a variety of things: the composition of the chyme, the acidity of the chyme, and the osmolality of the chyme. We've defined these three terms previously. I don't think we need to go over them again. Peristalsis is the reflexive movement or propulsion of chyme, generally anteriorly from the mouth to the butt, and sometimes in a retrograde fashion. It's a reflexive response. It happens if you touch the outside of the intestine, the wall will contract. If you climb inside the intestine, if you climb to do that and touch the wall of the intestine from the inside, it will contract there too. So what's that modeling? The movement of the bolus of chyme along the lumen. Mixing is very, very important. We talked about the importance of mixing in combining the gastric secretions with the chyme to ensure that there is a mixture, an admixture of enzymes, and also that anything released at the epithelium is brought into contact with all of the chyme. And then segmentation, we talked about this is important for ensuring that the digestive enzymes have access to all of the chyme by breaking it up. Through segmentation, the enzymes and the bile are able to access all of the ingesta. Now, in any standard textbook of GI tract physiology, you're going to find an almost endless discussion of intestinal reflexes. We're not going to get into all that. I just want to give you a couple of examples here, a few examples, and have you sort of work this out on your own. For example, there are intestinal reflexes. When the chyme moves into the intestine, that just means that the rate of secretion and motility is going to change. What you normally find in general, in not all cases, is that in a GI reflex, when the chyme, for example, enters the stomach, you may want to go to the bathroom. It's called the goose reflex, gastrocolic reflex, right? And what function is that serving? New stuff coming in, we've got to get rid of the old stuff. Gastroileal reflexes, the prefix tells you where the reflex originates. The chyme is present in the stomach, and there's going to be some distal intestinal response. As I say, elevated activity is generally going to accelerate the passage of the distal chyme, once again making a way for the new material. And then finally, this is one that you shouldn't be experiencing now, but you probably did in for lunch, something called the migrating myoelectric complex. This is the clearing motility. It often occurs on a schedule, especially with you folks eating at the same time every day, often occurs on a scheduled basis. That can occur anywhere from every hour and a half to every several hours. And it's often associated with borborygmus, that noise that your stomach makes when you're in that clearing motility, either immediately before a meal, or sometimes even after meals. But it is a type of clearing motility. And once again, it's taking everything in an anterograde direction, ensuring that new nutrient-laden ingesta can be taken in readily into the stomach and intestine. All right, let's take a break there. We'll come back and talk about the glands of the intestine.